Hello student, in the series of biochemistry, we have already studied about glycolysis, and you know that the glycolysis produce two molecules of pyruvate, and this pyruvate has different fate under different metabolic conditions like hypoxic condition and in presence of oxygen. We will discuss about it in detail. This topic is very important for those students who are going to take their competitive examination because this is the topic of examiner's choice. So let us begin with the fate of pyruvate. The pyruvate has three alternate fate under the different molecular metabolic conditions. So let us begin. The glucose yield two molecules of pyruvate by the reaction set of reactions we call glycolysis. The pyruvate then produced has three alternate choices to enter depending upon the condition of organism, that is under presence of oxygen and in the absence of oxygen. One of among them is ethanol fermentation, in which two molecules of pyruvate get converted into two molecules of ethanol and the production of two carbon dioxide molecules take place. This happens under the hypoxic or anaerobic conditions. And second alternate fate of pyruvate is the conversion of two molecules of pyruvate into two molecules of lactate. This again happens under the hypoxic or anaerobic conditions. Fermentation takes place in ethanol. Fermentation to ethanol take place in yeast-like organisms, and lactate formation is important in vigorous contracting muscles because muscles need energy. We will discuss it in upcoming upcoming slides. In R.B.Cs, in some other cells and some organisms, the pyruvate get converted into lactate and we call it as lactate fermentation. The very important fate of pyruvate under the organic or aerobic condition is two molecules of pyruvate get converted into two acetyl-CoA with the release of carbon dioxide molecules. The two acetyl-CoA then enter the mitochondria and goes into the Krebs cycle and produce the further four molecules of carbon dioxide and four molecules of water with the production of lots of energy. When there is enough oxygen, that is aerobic condition, the oxidation of pyruvate takes place. The aerobic organism, pyruvate is moved to mitochondria where it gets oxidized into acetyl-CoA. This process involves the release of carbon dioxide. Later, the acetyl-CoA is completely oxidized into carbon dioxide and water molecule by entering the citric acid cycle. This pathway follows glycolysis in aerobic organisms and plants. What happened to the fate of pyruvate under the absence of oxygen? Under the absence of oxygen, the pyruvate undergo anaerobic respiration or fermentation. We use this term interchangeably, but there is a difference. Anaerobic respiration is a respiration where there is no oxygen present. The last electron acceptor is inorganic molecule other than oxygen, wheat, sulfur or nitrogen or some other inorganic molecule. Keep in mind, inorganic molecule. But in case of fermentation, the final electron acceptor is always an organic molecule like alcohol or lactic acid. So we clear about anaerobic respiration and fermentation. So the fate of pyruvate under anaerobic respiration is first phase is lactic acid fermentation. In some organisms, pyruvate is converted into lactate by the enzyme lactate dehydrogenase. This reaction is reversible reaction, and it uses NADH2 and produce NAD. Keep in mind that NAD is very important because when the conversion of glyceraldehyde 3-phosphate into 1,3-bis phosphoglycerate, uh, NAD molecule is used which produce NADH2. If there is no NAD available at that point of glycolysis, the glycolysis could not go further beyond that point, and the cell will die because cell will not be able to produce energy. So in the absence of oxygen, uh, the cell is not be able to produce the ATP or not be able to oxidize the pyruvate, so no ATP will be produced and no NAD is produced in the Krebs cycle. So cell has to take the alternate path to generate these NAD. They either go for the alcoholic fermentation or they will go for the lactic acid fermentation. In the condition where the oxygen is insufficient, like the skeletal muscle cell, the pyruvate cannot be oxidized due to lack of oxygen. Under such condition, the pyruvate is reduced to lactate by the process of anaerobic glycolysis. Lactate production from glucose also occur in other anaerobic organisms by process of lactic acid fermentation. Another fate of pyruvate under anaerobic condition is the alcoholic fermentation. In some microbes like yeast, the pyruvate formed from glucose is converted anaerobically into the ethanol and carbon dioxide. This is considered the most ancient form of metabolism of glucose. In this process, the pyruvate get converted into acetaldehyde by the enzyme pyruvate dehydrogenase. Pyruvate dehydrogenase uses the thiamine pyrophosphate, which is derived from the vitamin B1. With the release of carbon dioxide molecule, the acetaldehyde produced from pyruvate get converted into ethanol by the enzyme alcohol dehydrogenase. This reaction is reversible in nature. That means ethanol can be converted into acetaldehyde and acetaldehyde into ethanol. This reaction uses NADH2 molecule to produce NAD positive, which is used in the glycolysis as I told you earlier. With this, alcoholic fermentation will take place. Interesting scenario occur in case of livers, in case of skeletal muscle cells, because skeletal muscle cells require a lot of energy while doing work. Skeletal muscle continuously contract. In case of when you are working in a gym or when an athlete is running, their skeletal muscle need the continuous supply of ATP in the energy in the form of ATP. What skeletal muscle are under hypoxic condition, that is the concentration of oxygen is drastically reduced inside and muscle cells during the vigorous activity. So what happened? Glucose is converted to lactate by lactate fermentation. This lactate is transferred to the liver where it gets converted into glucose again, and the process is known as gluconeogenesis, and glucose then again transported to the muscle cell where it goes under the glycolysis and produce more energy. Lois Pasteur observed a very interesting fact about glycolysis. Is he observed that under the aerobic condition, the glucose consumption and ethanol production is dropped. This is because you know that on complete oxidation of glucose under oxidative phosphorylation yield 32 ATP per molecules, and under the fermentation we get only 2 ATP per molecule. So what we get, and we know that the dividing cells require a lot of energy, so they have to be consume more glucose so as to compensate the energy requirement. And whereas the organism who are growing under the aerobic condition, they have enough of energy, so they need not to consume more glucose. This is known as posture effect. This slide is very important for the student who are going to take their examination, because this topic is very important for the examination point of view. And here we will discuss about the energetics of glycolysis. In glycolysis, the formation of 1,3-bisphosphoglycerate from glyceraldehyde 3-phosphate yield 2 NADH molecules, which further go into the ETC chain and produce 5 ATP, because one NADH molecule produce 2.5 ATP. In second step, there is a formation of 3-phosphoglycerate from 1,3-bisphosphoglycerate. This is known as substrate level phosphorylation, and it produces 2 ATP. Other point where the ATP production will take place is the formation of pyruvate from phosphoenol pyruvate. Here again 2 molecules of ATP are produced. So the total ATP production is 5 from NAD, 2 from 4 from substrate level phosphorylation. So the total 9 ATP are produced. What? In preparatory phase, or in preparatory phase, the 2 molecules of ATP are actually used by the organism. So when we subtract this 2 ATP from the 9 ATP, we are left with 7 ATP. So we can conclude that under aerobic respiration we will get 7 ATP as a result of glycolysis. What in case of under the anaerobic condition, when fermentation take place, you know that this NADH produced in the reaction is used for the production of NAD. So the 5 ATP which can be produced from NADH are not available, so we are left with only the substrate level phosphorylation, which produce 4 ATP, and when we subtract 2 ATP in the preparatory phase of the glycolysis, we are left with 2 ATP at the end. So we can conclude that under the aerobic respiration we will get 7 ATP, and under the anaerobic respiration we only get 2 ATPs. So there are some frequently asked questions about glycolysis. Pause the video and try to answer this question. What is aerobic glycolysis or respiration? The aerobic glycolysis is the process of oxidation of glucose into pyruvate, followed by oxidation of pyruvate into carbon dioxide and water molecule in the presence of sufficient amount of oxygen. The second question is, what is anaerobic respiration? Anaerobic respiration takes place in the absence of oxygen, resulting in the reduction of pyruvate into lactate or alcohol and reoxidation of NADH into NAD positive. Where does glycolysis occur? The answer is glycolysis takes place in the cytosol of the cell. What are the product of glycolysis? The product of glycolysis are 2 moles of pyruvate. What we get at the end of glycolysis? We get 2 molecules of pyruvate, 4 molecules of ATP. Net gain is 2 ATP molecule and 1 more molecule of NADH. Our next question is, how many NADH are produced by glycolysis? The answer is 2 more molecules of NADH are produced by glycolysis. How many ATPs are formed in glycolysis? A total 4 molecules of ATP are formed in glycolysis. The net gain of ATP in glycolysis is just 2, as 2 ATPs are utilized during the preparatory phase of the glycolysis. What are the function of glycolysis? The primary function of glycolysis is to produce energy in the form of ATP. Similarly, glycolysis also produces pyruvate, which is then oxidized further to create more ATPs. One more thing before we conclude glycolysis, we must know that glycolysis is very important for the brain cell because brain cell cannot undergo Krebs cycle, so they are solely depend on glycolysis. And other is that the R.B.C cell, mature R.B cell don't have mitochondria, so Krebs cycle don't take place in mitochondria in RBCs, so they are also dependent on glycolysis. With this, we conclude this video and we shall meet in next videos. Thank you.